My name is Shekhar Seshagiri. I'm the chair of the NGBD 2014 meeting. On behalf of the organizing committee, I want to welcome all of you. Many of you come from various parts of India and also from various parts of the world. We will kick off the conference with inauguration by the Vice Chancellor and Director of NIMHANS, Dr. Satish Chandra. So may I ask Dr. Satish Chandra to join us on the stage. I would like to invite Dr. Akhilesh Pandey, the co-host from IOB and the co-chair, to join us. Now I'd like to request Dr. George Thomas, who is the co-chair and co-organizer of the meeting, to join the stage from SGRF. And then I'd also like to request Dr. Andrew Peterson from Genentech to join us on the stage here. Take any of the seats. I'd also request our keynote speaker, Dr. Stefan Schuster, to join us on the stage here. I'd also request Dr. Mitali Mukherjee, our previous conference host, to join us on the stage. So we'll kick off the conference with a ceremony to light the lamp as soon as Mitali is. I guess there's no seat for you, huh? We'll find the seat. No, there is. There's one more chair. Yeah. That's okay. So do we have first Dr. Satish Chandra, Akhilesh, Stefan, and Andy to light the lamp, and I guess.
Vatapi Ganapatim Bhajegam 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 Varanasyam Varapradam Shri Varanasyam Varapradam Shri Varanasyam Varapradam Shri Vatapi Ganapatim Bhajegam Varanasyam.
Give them the small mementos. So, this is the fourth year of this meeting. The meeting started in 2011 in Trivandrum. Then the subsequent year, we did the meeting in IIT Chennai, and last year, with Mitali's help, we had it in Delhi. And this year, it's actually a much bigger event. And when we came and looked at NIMHANS, it was such a perfect venue. And the director was, the vice chancellor was very kind to work with us to get this venue organized. So I'll give you a little bit more detail about the meeting after the hosts have a chance to introduce themselves. So, oh, is that right? Okay. Then I'm going to have to connect my computer in here. So we have over 80 talks spread over 38 sessions, and there's 3 concurrent sessions, and there's 8 keynote talks that will happen in this venue here. We also have over 80 posters that will happen on the first floor. We're also doing some real-time genotyping studies. I'll go over some of that, and there's also multiple exhibitors outside. We already had 2 workshops, one on proteomics at NARA in Italia, and another one in Institute of Bioinformatics for the last 2 days. There's also a job fair arranged for tomorrow. And in addition, to bring science and art together, we have an art exhibition that's right outside the venue here. And there's also, George will make some announcements about some awards and scholarships. This all come together as part of the conference. But the main objective of the meeting is to bring together people who are interested in science and work in the area, and then also to bring people who want to learn more about the area and interact. And the objective is to create an ecosystem where people can come together, interact, and learn, and take the knowledge back to their science. So maybe with that, I'll stop, and I'll invite Dr. Satish Chandra to talk about NIMHANS as the co-host.
Good morning. Dr. Akhilesh Pandey, my friend, and from the Institute of Bioinformatics, from Johns Hopkins, with whom we have NIMHANS as association for more than nearly a decade now. And we are working together for a decade now, along with the dignitaries on the dais. I also know Maithali very well for many, many years now. And we are all assembled here for a very important conference, which is talking about the next generation genomics. So we all know that the Genome Project draft was completed in 2001. It is about nearing now 15 years. And it is a time now to take stock of what has happened after the Human Genome Project was drafted. I'm sure you could recollect when the Human Genome Project draft was published, the people were wondering what does it mean and what is it going to take place further. As we can see in the last 15 years, there is a lot of advancement which has happened. The most important advancement following this Genome Project was in the field of technology. The technology has advanced so much that now the people can do the same work which they were taking years together to complete, could be done in a very short period of time. And the technology has advanced so much that the large genome projects could be taken and completed in a very short period of time, be it a plant genome or an animal genome or a human genome project. At NIMHANS, we are working on genetics for quite some time. We started with the usual way, as anybody does, starting of the SIP pairs, looking at the families, and then finding out a genetic association. I myself, being a clinician, a neurologist, was interested in the field of epilepsy, started working on epilepsy genetics in collaboration with a neighboring institution in Bangalore. When we started looking at the families of epilepsies, and we were able to do a genetic linkage studies, and then we said, there is no point in just doing the genetic linkage studies, and now we need to work further. It is easy to find or do already which is known, gene is known, so you can translate that information to the clinical aspect. That is being done now for the diagnostics in many of the centers. But what we wanted to look at is some of the rare conditions wherein we have problem in finding the gene associated with that. And one of the study which I myself was involved was with the consortium, the World Consortium on Lafora body disease. And this is a rare disorder, as many of you may know, and this rare disorder had a problem to find out the genetic association, as the mortality in this disease was almost 100%. And we worked on for a very long time, and then with the consortium from all over the world, we were able to find a gene. And that was an epilepsy myoclonus gene, EPM2A. And that gene has been published in the Nature Genetics in way back in 1998. And after that, we have our own problems in this country, and for our problems, I don't think anybody else will work for it. And we need to find our problems and solutions from our side. From that angle, we started looking at the genetic association with a type of epilepsy, what is known as hot water epilepsy, which is widely prevalent in this part of the country. And when we started working on that and looked at the relationship between this condition and various other genetic association, because we understood that it was a hyperthermic condition which is responsible for this type of epilepsy, what we find in this part of the world. However, we didn't know what is the mechanism, and we traced the mechanism with an animal model, and following that, we did work on to find out whether it is similar to febrile convulsion gene. But what we found was it was not, and hence we had to go for the whole genome scanning. And that was the only way we were able to look in this entity. And I were very happy to say that we are able to get a gene for that. And that is now being sent for publication. And we have an association which has already been published in the Human Genetics, Human Molecular Genetic Journal. But what is important is this required a whole genome scanning. And that facility was developed in this institute and also in the neighboring institution, and that's why we were able to move forward or march forward. So now in this institute we have a molecular genetics laboratory, and we are working on various conditions, some known disorders, some non-unknown disorders. And one of my colleague, Dr. Satish Girmaji, I'm sure he will be talking about it in the later part of this conference, has also isolated a gene for the first time for a mental retardation and microcephaly, which has already been published. So these are the discoveries which have been done from this institute. Taking this further, now IOB is working very closely with us, and in our Neurobiology Research Center, which has been developed in this institute, we have a space for IOB. And IOB lab is working very closely with NIMHANS for the last 5 years. And what could be achieved was that we have a draft human proteome, which has been published in this year in the Nature on May 29th, which is one of the major discovery, I should say, and which has received a worldwide recognition. And that is possible because of the work from IOB along with Johns Hopkins University and the NIMHANS. So that is possible now, and we have recently a new machine for the proteome, which is an Orbitrap 1, which has been introduced in the institute. And with this, we thought that our work on proteomes will go further in collaboration closely with IOB. So neurological disorders are rare disorders, which are classified as rare disorders, and we have many disorders we are still to be questioned. We do not know how it is caused, why is it caused, and unless and until we know how and why, we cannot manage these patients. And that is the problem as a clinician we all face. Whether it is a systemic disorders, whether it is a multi-system disorders, neurodegenerative disorders, there are plenty of them which yet to be answered, which is a fertile ground now, and this area has to be probed now. And being a mental health and a neurological institute, the NIMHANS has got interest in these areas to find out what are the causes for these disorders. And hence my colleagues from psychiatry as well as from neurosciences are working towards finding out the etiological causes as well as the genetic relation between various neurodegenerative or psychiatric disorders. And since these are complex disorders, naturally the work is not so simple and needs a lot of work and a lot of the geneticists to work together. And I'm happy that this next generation genomics conference, which is being arranged now today in the NIMHANS campus, will boost many of the people and also the scientists who are working in this area to learn more about it and to see how the present work can be taken to the next level, which is very important now. And that is the only way we can march ahead. And that is what we are now looking for, the work on genetics to take a next step and then see what India can contribute for the world genomics. I wish you all the best, and at the outset I welcome all of you who have come from far off places, including abroad, to Bangalore, and which is a very nice climate now in Bangalore, and I'm sure you will enjoy the climate as well as some part of the garden which is left, at least in some part of the city, though it is called a garden city. So at least you go around and enjoy yourself, and you have a very good scientific feast in the next 3 days. Thank you very much for this opportunity.
Thank you, Dr. Satish Chandra. May I ask Akhilesh Pandey to say a few words about IOB and
Professor Satish Chandra, Director of NIMHANS and Vice Chancellor, Dr. Andy Peterson, Dr. Stefan Schuster, Dr. Mitali Mukherjee, and Dr. George Thomas, speakers at this meeting and participants. Welcome to you all and good morning. I think this is a very, very exciting meeting on next generation sequencing, and the NIMHANS, Institute of Bioinformatics, and Sai Genome Research Foundation are the three co-organizers of this meeting. And there is now a NIMHANS IOB lab within NIMHANS, which has been the basis of a very strong collaboration on proteomics, but now increasingly on bioinformatics, but also on genomics. And next generation sequencing has really brought about a transformation in the field of biomedical research. And over the course of this conference, you are going to hear not just about biomedical applications, you hear about bioinformatics and tools, analysis methods, and applications to other fields that we are not normally talking about in biomedical scenarios like agriculture. And what you'll also hear about is some very hot off the press data that has resulted from a very nice collaboration that IOB has had with NIMHANS, where Sai Genome was involved, where a new mutation has been found for X-linked mental retardation. So we have just completed this, and I think I would like to thank Sai Genome for setting up this type of a facility that will allow investigators to actually do, as Professor Satish Chandra pointed out, things at the whole genome level and the exome level, in addition to the other smaller things that we have been doing all along. And I think this 3-day conference will be an exciting one to watch, and I'm really looking forward to all of the speakers talking about the latest advances. And all of these speakers, what is common to them is that they are all at the top of the field. And
of course, Dr. George Thomas to introduce SGRF.
Hello and good morning. Just a brief intro about SGRF. Sai Genome Research Foundation is a not-for-profit organization. We are into research and also community outreach programs. We are into science education, and this conference is part of our science education activities. As Shekhar mentioned, we had 3 NGBT or next generation sequencing conferences so far, and this is the 4th one in Bangalore. Now in terms of research, we try to do work on medical genomics, on agricultural genomics, and also on environmental genomics. So the various projects are covering these areas. I had a small presentation, but because it's a little difficult to project now, I'm speaking without the help of the slides. Now we also have this encouraging researchers and students program. So as part of it, we have given some awards to the faculty of Kilikulam Agricultural College, and also sponsored 2 students from that college for their education. And today in this conference, these awards will be presented. This year we have instituted the Excellence in Science Awards. So we accept nominations from various researchers, and then a committee screens it and evaluates. So I'm very happy to announce this year's winners. Actually there are 2 winners. The first one is Dr. I.C. Verma from Sir Gangaram Hospital, Delhi, for his significant contributions towards bringing medical genomics to the patient. The second winner is Dr. Mohan V. Mohan of MDRF, Madras Diabetic Research Foundation, who has done path-breaking research into the genomics of diabetes. And I wish to congratulate the two winners, and also want to announce that this is an annual program, and all of you are welcome to make nominations for the next year's Excellence in Science Awards. Now, I welcome you to this meeting. I hope you have a nice time. Please enjoy and benefit from this meeting. And because of time shortage, I also want to express at this point my gratitude to all the dignitaries on the dais who had agreed to come here and grace this occasion, and also thank all of you for coming from so far away in many cases and making this a success. I also appeal to you to provide feedback on this conference so that we can improve upon this for the future conferences. Now,
Thanks, George. So I think this sort of concludes our session on the conference inauguration. So I want to thank all the dignitaries on the stage, and as you exit, we would like to share a small artwork done by one of the artists, Dr. Geeta, that's displaying her artwork here. This is the chariot from Hampi as a recognition of the architecture marvels in Karnataka. So I'll ask George to present as you guys exit down to the podium. Andy, maybe we'll start with you. Yeah, George. Okay, so we'll now start off with the official program. So what I'm going to do is take literally 5 minutes to go over the conference-related stuff. Can somebody take that? Miller. Yeah, thanks. Okay. Yeah. So we'll try and make up quickly for the time. Switch over to my. Can you switch over to my slides? Okay, great. So once again, thanks to everybody for assembling here in Bangalore. So as we said, this is the fourth meeting in the series. So there are over 80 talks spread over the 3 days, 38 sessions. That's okay. And we have some fantastic talks, including talks from two of the SGRF award winners, and also a special message from Randy Sheckman, the 2013 Nobel laureate. On behalf of eLife, Dr. Walker will be talking tomorrow right here in this venue. So all the keynote sessions will happen in the venue. They're arranged in a way where everybody can get back together every few hours. We have over 80 posters. These will be displayed throughout today and tomorrow, also on the third day. And also to encourage students to be more participatory, we'll give them a chance to pitch their posters 9 o'clock, 9 to 10 tomorrow and the day after. And you can turn in your slides by end of today, 2 slides, 60 seconds. And many journals, eLife, PLOS, JCS, Nature Review, FEBS, and EMBO have all donated money to the conference to support the poster prizes. We'll also be doing a real-time genotyping. I'll tell you about it in a second. As I told you, multiple vendors. The workshop's already done. The job fair, an art exhibition. And throughout the conference, you'll see some awards and scholarships being announced. So quickly about the real-time genotyping. Last year at NGBT in Delhi, we actually sequenced mitochondrial DNA. We collected samples from consenting patients, and the DNA was isolated, and we sequenced it in a matter of basically 3 days. PCR amplified the mitochondria, made the libraries, and we actually sequenced, collected the samples. The sequencing happened in Cochin. The analysis happened between Singapore and Pennsylvania, and we were able to upload the data. It was a huge collaborative effort, and in fact, by the third day, we were able to show the genotype of 80 participants at that meeting. And as you know, mitochondrial haplotypes are used to track human migration and many other things. But the neat thing about this was it was the first time that you could actually do it in a conference in a very short order. The thing that we proved was this was truly an international conference, and also the M haplotype was the major one. An expensive way to do it, but at least showcase the technology at that point. So this year, we took a slightly different tack. Thought we'll do it a little bit more fun. At about 12:30, we'll start collecting samples in the first floor. People can go there, and there'll be a little bit of education about what will happen. And the sample collection will happen throughout the day until about 6 o'clock. So there'll be plenty of time. The idea is you sign a consent form after reading it. The cheek swab is taken. Regions of interest will be amplified, and then using a mass spec-based nucleic acid mass spec, we look at a few SNPs, and we call this Varetime VARMAP project. And it's actually, again, starting today, and by this date is wrong, on the 19th, we'll actually have all the data, and we'll announce the findings. This is done in collaboration with actually Madras Diabetes Research Foundation, where we'll collect the genotype data, and the MedGenome folks, along with colleagues from Genentech, are providing some of the technology support that's needed. And the SNPs we'll look at are 5 diabetes risk SNPs. This doesn't mean one is going to get diabetes, but it'll tell you whether you have a risk. And then some fun ones. One is called the cilantro or coriander. Some people cannot taste it, or when they taste it, it's soapy. There's a SNP in a particular gene associated with that. And then another SNP associated with smell. Enones are known to be flowery or citrusy compounds. They're actually put in many scents and so forth. But some people cannot smell it at all, and that's to do with one variation in ABCC11. And very similarly, there are some people who don't have body odor, and predominantly a lot of Asians, and that's to do with a mutation or a variation in a gene called RB5A. So we'll type these, and later on, at the in a couple of months, you can actually download your own data and do whatever you want with it. But we'll provide aggregated data at the end of the conference. This is again to showcase the technology. So some housekeeping stuff, very quick. I'd ask all the speakers to try and stay on time. There is sufficient help in the auditorium, your chairs, the instructions. They will run the show. They have session managers in every hall. They'll help and get all the AV stuff. And then an important point, please put your phones in silent mode, and if you need to speak, be polite and walk out and use your cell phones. There is a help desk right in the front, and they'll assist you with whatever you want. We're trying to do our best. It's not going to be perfect, but if things are not working, feel free to let us know. We'll try and fix. So as I said, the help desk is right outside Auditorium 1 as you exit to your right. The poster sessions, I already told you, posters 1 to 40, you need to be by your posters between 6 and 7 today, and 7 and 8, 41 through 80. The lightning rounds will happen on the 19th, 18th, and 19th. First 1 to 40 tomorrow, and 41 to 80 the day after. There's lunch, tea, there's food. As you exit the auditorium, main auditorium to your right, there's a very nice eating area. There'll be food. There's also a mixer tonight at the first floor where the posters will be on display, so feel free to join. And in fact, the inauguration session, the first session, is open to all the faculty and staff at NIMHANS. We've also opened up one of the clinical sessions and also a session where the NIMHANS faculty are speaking. So please ask your colleagues to join us at this meeting if they can find time. We're quite grateful to many other folks that have provided a lot of support, particularly the poster prizes, the eLife, Nature Cancer, JCS, FEBS, PLOS, and also EMBO. And more importantly, running this conference actually does, believe it or not, cost money. And so we're very grateful for some of these sponsors that have provided the support, and particularly Illumina and MedGenome, the LifeTech, PacBio, ILS, EMC, Isilon, IDT, Roche, EA, Quintiles, New England Biolabs, AbsciX, and Agilent Technologies. There's also other partners, BioSpectrum, India Biosciences, they're not on here, but Able, for their support with a lot of the other logistics and things. So with that, I'm going to stop, and I'm going to invite Dr. Stefan Schuster to kick off the first talk. As Stefan gets up here and hooks up his mic, I'll probably say a few words about him.